is everybody today? Hope you guys are doing stressed. Yeah, I hear you on that. Okay. I was joking with Dr. Merle that cracked a tooth. I said it was the stress of grinding my teeth. Okay. So, why is that right in the middle, of course? To pick up where we left off, we're coming up to the end of chapter 3. We'll probably finish up everything I want to talk about on Thursday. We'll do a little review session next Tuesday and have our first exam the following Thursday. So I think that's the 10th, I believe. Thursday. Yes, yeah. Is that a bad day?
I mean, I just seem kind of bad. It's like,
yeah. Well,
so we're gonna come to class to do that exam?
I will figure that out by next week. Hopefully by Thursday, but yeah.
So we're off Monday. And then we have class
On Tuesday.
We are off Monday. So we have a test on Thursday.
Right, yes. Yep. Okay.
That's all we were doing.
But as to what we were talking about amino acids in particular and how they have functional groups that function as both acids and bases. You got the amino group and you got the carboxylic acid group. Both of those are ionizable. And in the case of histidine, the R group also has an ionizable subunit here as well. So if we take a look at our histidine, this is what I actually kind of recommend, kind of writing this out. And then I'm going to try and do, I might do that on Thursday. But I'm going to try and draw one out. And I'll show you, if in the case of like histidine, this is a good thing to do because we draw the whole structure there and you can see everything. But if I give you a small peptide, a lot of times the only thing we need are the ionizable groups. We'll talk about that. So if we put this in the most acidic solution we can, everything's protonated. You can see you've got this NH3 plus here, this NH plus here, and our COOH just has no charge. So our total charge is going to be +2. The first proton that comes off is pretty much always going to be the carboxylic acid group. All right, so when this one comes off, we raise our pH a little bit, abstract this proton. Now we have a negative charge here, but we still have our 2 positive charges. So our total charge on histidine is now +1. All right. And our pKa for this carboxylic acid group, pK1, is going to be 1.82. So we can make a buffer solution here. But I've also gone and made this +1 in this area. Right. If I keep going, the next proton that's going to come off is going to be in the R group. So our pKr is 6.0. That's when this proton starts to come off with our nitrogen group here. So here, when this one comes off, I'll now have a plus charge for the amino group, a minus charge for the carboxylic acid group, and no charge here. So my total adds up to 0. So when I get to isoelectric point, this is going to be one of the ones I want to use. Okay. When I look at this, if I keep going and make it more basic, I'll finally abstract away the amino proton. All right, and this pK2 is at 9.17 for histidine. So once I abstract that away, I'll now have a neutral charge, and a negative charge. So I've gone from 0 to -1. All right. So if I want to know my isoelectric point for histidine, I'm just going to average the 2 pKa values that go from +1 to 0 and -1 to 0. In this case, pKr and pK2. So if I add these two together, I get 15.17 divided by 2, and I get about 7.6. Okay. So not awful, right? And I like this figure because this packs in a lot of information. I think I left off with this figure, but we have our graph of the pH over time. As you can see even though these don't end up being pure plateaus, purely flat, you can see little areas where the curve is not too great. So these are the 3 regions where I can make a buffer solution. When I used to do, well, actually almost every year except for this year, when I do the titration of the amino acid in there, you end up making a graph like this. And in a perfect world, you get something that would look like this. I would give groups that wanted to challenge histidine to see if they could actually resolve all 3 of these. What tends to happen is when you have amino acid groups that have an ionizable side chain, it often kind of overlaps with another. For example, if we looked at something like aspartate or glutamate, their PK1 and PKR are like half a pH unit away from each other. So really what you get is kind of one long curve there with maybe a little bump in it. But histidine, theoretically, you should be able to resolve all these. It is still challenging to do, but it's possible. But the idea, again, we have kind of our little plateaus where we could have a buffer solution, and you have our places where we're far from that, where we're going to see a small change of acid or base added that's going to result in a big pH change. And of course, we have our isoelectric point here where our Zwitter ion form is made. I can't remember if I mentioned that on Thursday or not, but our Zwitter ionic form is where we basically have these stabilized ionic charges, but the net charge is 0. So I've got a +1 and a -1 that cancel each other out, but the overall charge is 0, so it's not going to move in an electric field. If we looked at a three-dimensional structure, it'd probably line up in an electric field, which might be useful for maybe trying to make crystal structures at some point, but that's getting way beyond what we want to talk about here. All right. To look at this in words, maybe, come on. Come on. There we go. All right. Just have to hit the button with authority. So look at this as words. What we really want to do to figure out that isoelectric charge is find out two, well, I guess really three, because you go from +1 to 0 and 0 to -1. Or if you like to do it the other way and start with everything abstracted and just add protons where they can fit in, and you can go the opposite direction as well too. All right? And then find out these species in here where you have a positive charge and negative charge. Look at those two pKa's, and those are the ones that you'll average. So if we had, a peptide that had 150 amino acids hooked together, you might have maybe 50 ionizable side groups on there. Right? So maybe if it's fully protonated, it might have a charge of +25. And if it's fully deprotonated, it might have a charge of -25. The only ones we care about are going from 0 to +1, 0 to -1, because that's where we're going to balance it. So I might have, thousands of pennies and nickels, and as long as I balance them on here, there's only one spot where I move from one side to the other, it's going to balance. All pennies, maybe. Treat our protons as pennies, because no one cares about it. Toss them out in the parking lot, right? It's terrible. All right. Any questions about the isoelectric point and how to calculate it yet? So I think I've made available an example, maybe two examples of how to calculate it for a small peptide. I'll actually probably go over one of those either on Thursday or Tuesday as well in person, because that's something I'll probably be asking you to do. And there might even be a short video of someone doing it. I think the short videos of derivation of the internet. I don't think, yeah. Maybe it'll be me. Maybe I actually watched my own Zoom video for the first time after posting like 50 of them. I'm like, hey, cool. I don't sound like a total goober. This is okay, aside from all the pauses. All right, and random stories that have no point, like this one. Well, they have a point, but All right. So moving on, we've been looking at individual amino acids, okay? And all of them have at least two, and some of the side chains have a third ionizable group. But where they get really interesting, much like if we have our little Legos, we can hook them together and make something bigger, okay? So our amino acids are going to polymerize. And actually, we did get through a little bit of this. The key here is, repetition is good. We're going to have this dehydration, condensation reaction. We're going to remove water. We're going to move the OH group from the carboxylic acid and the proton from the amino group, and we're going to end up making this peptide bond, okay? And the crazy thing is we're going to talk about the peptide bond in like 3 or 4 different places. So peptide bond, big important key. If we hook a bunch of them together, you can see we have this we're constantly making this little peptide bond between our different R groups. So our R groups are different decorations, and the peptide bond is a constant. And then we'll have this amino terminal and carboxy terminal ends. And then for this one, we would number this. We number from the amino terminal end. For this one, we have serine, glycine, tyrosine, alanine, and leucine. We could give it this big long name, we could give it this short abbreviation, or we could just call it SGYAL. So I always joke scientists are lazy, efficient. We want to find the way to give as much information as we can in the least amount of words, effort, stuff like that. Okay. So you got to know those single letter abbreviations. So once we start actually connecting amino acids, we form peptides. Sometimes we'll call them oligos. There's this whole, variable. It's like, well, it's kind of like, what do we mean by small in chemistry? And it's like, well, we talk about small molecular weight for organic chemistry, and you're talking maybe a few hundred grams per mole. Small peptides, we're talking maybe 10,000 grams per mole. So if we have just a few peptides, we might call it an oligopeptide or a short peptide. Depends on if it's active or not. A lot of small peptides are used to trigger different effects. Insulin is a small peptide. I think its pre-pro protein has 56 or 57 amino acids, and a bunch of those get trimmed out. So its active form is only maybe a couple dozen. Oxytocin, this is like the new baby smell, which is key for bonding. This is one of the few known human pheromones. There are actually studies that people who are oxytocin deficient won't recognize children when they're born as their own children. It's crazy stuff. There was also, I think back in the 90s, maybe early 2000s, a perfume company that put oxytocin in a perfume to ideally try and make people feel good, presumably about themselves, but the whole idea of pheromones attracting other people. Or something like that. We'll see some neuropeptides. Substance P is a big one in pain mediation. So it tells you how well you feel pain. If you look at, say, like the opioid epidemic, it's a major concern across the country. Finding ways to treat pain without an addictive painkiller, probably a good idea. Various antibiotics, look at like polymyxin B or bacitracin. You ever got like your cut and you put a little probably bacitracin is one of the things, one of the common over-the-counter ones there too, to prevent bacterial infection. These are antibiotics that will kill gram-positive or gram-negative bacteria. Various mushrooms will have like a manatin, I think it's the death's head mushroom, angel of death mushroom, something like that. But a manatin is a horrible neurotoxin for us. Basically, it developed so that animals wouldn't eat the mushrooms. They recognize those mushrooms and say, don't eat those, you will die. Cone snails or scorpions have these other toxins in there as well too. And all these are just examples of small peptides. Right? If we keep putting peptides together, we can make large proteins that will have more complex functions. But these often play a role, like say, in signaling or organismal health or protection. Okay, so a lot of mediation in there. Okay. So what's the difference between a peptide and a protein? Well, it's really size, all right? So our proteins are composed of polypeptides, basically long chains of amino acids that are hooked together. You might have multiple chains in there as well too. When we get into chapter 4 and talk about protein structure, we'll see how that all works out. But our polypeptides are basically covalently linked alpha amino acids, and they may contain other stuff in there as well too. So a peptide is only amino acids. Our protein might have cofactors, like functional non-amino-based organic compounds, say like coenzyme A or something like that might be in there. Well, sorry, coenzyme A I guess would be our coenzymes, NAD plus and stuff like that. Maybe ATP would be a cofactor. We can have prosthetic groups, which are covalently attached or sometimes not covalently attached, like heme and myoglobin. And we can have other modifications that are on there as well too. So basically our proteins are a little more complex than just peptides. They don't have to be, but they can have all this other stuff in there. If I want to do definitional type stuff, I might say, hey, what's a cofactor? What's the difference between a cofactor and a coenzyme? All right, so our coenzyme, really, they overlap. Our definitions can kind of overlap a bit. We also, there are 20 amino acids that you should know, but there's more than 20 amino acids. There are uncommon ones out there as well too. And these are ones that are not recognized by the ribosomes. Our ribosomes only recognize the 20 basic ones. Although there's some little exceptions out there. There is this selenocysteine. It's a cysteine that has a selenium ion instead of sulfur in there. That is recognized a very minor bit by ribosomes and plays a minor role. But there are other ones that just aren't recognized and are incorporated as well. But they arise due to post-translational modification. So we might incorporate maybe an alanine in there, and then another enzyme comes along and changes it, makes a substitution or does something. It's post-translational modification. The analogy I always like to use is when you buy a car off the dealer's lot, and then you put a wing on the back and give it spinning rims, and put the underlighting on there so it's purple, and I don't know, something make your super cool car. Terrible, right? But those are all aftermarket modifications. You can do the same thing here as well too. Some of these are reversible, some are not. But a lot of the reversible ones, maybe we'll phosphorylate it. We'll basically add a phosphate group to, say, our tyrosine or serine or threonine, because those hydroxyl groups on those side chains make good targets for phosphorylation, and that can be useful for cell signaling and regulation. Kind of functions as a light switch, turn on or turn off a particular function. Do you need to memorize these? No. Oh God, no. Because there are so many. But we can start with something that's relatively simple. We have this 4-hydroxy proline. We just have a proline, but we stick a hydroxyl group on it. Okay? That's just another modification in there. Same way we can have this hydroxylysine or methylysine. So we can add either a hydroxyl group or a methyl group. Notice we add them in different places. There's a carboxyglutamate. Right? So we just stick another carboxylic acid group on there. A desmosine, we start attaching them around in a linked ring structure. And selenocysteine has selenium instead of sulfur in there. Obviously, we're not going to make disulfide bonds. You could theorize, well, can we make diselenium bonds? Maybe, but and then you can have even like pyrrole groups in here, and you can add more and more complex stuff. We'll also see that besides adding, groups like this to it, we can also add carbohydrates or lipids as well. And we'll talk about N-linked and O-linked, which are just what they sound like. We're going to attach to an oxygen or a nitrogen for our carbohydrates. But that's more in like chapter 7 or 10. 7, I think. All right. Reversible modifications. Again, any type of phosphorylation. We can have a phosphorylase and we can have a kinase to either remove it or add it. Okay. Adenylation is another one. We might add an adenosine group to it. In this case, a phosphoadenosine. And again, some methylation, like our glutamate methyl ester in here, is reversible, and our acetyl group would be reversible as well too. Again, these modifications, there are so many modifications. It's kind of like if you go to a car show and look at all the ways that people have, played with their cars to make them better or make them prettier or give them a particular function. You ever see like the video of the guy who turned his lawnmower into a tank? I think some guy in England who, not an actual functioning tank, but he put a little turret on there and and decorated it so it looked like a World War II battle tank, so he could go mow his yard and scare the neighborhood kids. Or it's like the but that'd be an example here. It has a specific role to play. Our polypeptides, when we talk about our polypeptides, they can be of a huge amount of difference in sizes. If you look at something like cytochrome C, that's probably one of our smallest proteins that we will talk about. It's got only about 104 amino acids in it. Use amino acids and residues interchangeably here. So it's got a molecular weight of about 12,000. So basically, if you want a quick estimate of the molecular weight of a protein, you kind of take the number of amino acids and multiply by 110. That's a good average. Right? So that'll get you in the ballpark. Okay? You can see something like ribonuclease A, we've got 124 in here. So 110, this would be something like 11,000 something, close to there. But we look at this, we can get larger ones. Our lysozyme, myoglobin from the equine heart. Hemoglobin is about 4 times larger than myoglobin, which makes sense because hemoglobin is essentially 4 myoglobins kind of hooked together, sort of. We'll see that in the chapter 5, I believe, comparing those two. Serum albumin in here, they're all maybe like our average size ones. We're in the upper tens to lower hundreds of thousands. We go down here, Titan has a molar mass of almost 3 million grams per mole. Okay? And this is a protein isolated from humans. I believe it plays a structural function somewhere in there as well too. But if you wanted to collect 1 mole of Titan, you'd have to grind up a lot of humans. Yeah, you need like what, almost 3,000, almost, that's per mole. So that would be, yeah, you need about 3,000 kilograms. And this is not like a super common protein. So if you want like 1 mole, a 1 molar solution here, make a liter of 1 molar solution of Titan, which I don't think you do, but you'd need a lot of Titan. Grind up a lot of people. Make soil like green. It's people. All right. Conjugated proteins end up being covalently bound to something that is not a protein. So we can take protein pieces and hook them together, but conjugated proteins are going to have something that's not protein. So we can have our lipoproteins. We'll talk about those in Biochem 2, particularly our cholesterol metabolism. Our lipoproteins play particular roles in transporting lipids and fat in the cholesterol. Glycoproteins are going to have carbohydrates, glyco for glucose conjugates in there. So things like IgG. We have our phosphoproteins, we'll have phosphate groups on there. Hemoproteins will have heme. Flavoproteins are my favorite because I get to say flavin. Our flavin nucleotides will be attached to those. And then of course we have our metalloproteins as well too. All right. So when we start talking about proteins, there's a lot of questions we can ask. All right. So it's like, wait a minute, aren't we supposed to get answers here? Nah, now we get questions. So when we look at a new protein, we might say, well, what's its sequence? What's its amino acid sequence? That'll be our primary sequence in there as well. Primary structure. What's it made of? Okay. What shape does it take? Is it a long linear protein like myosin or actin, or is it a big blob like hemoglobin? How does it get to its native fold, its native shape? Does it require chaperones? Does it spontaneously fold without anything in there as well too? Does it require some energy to make some of these particular folds? Does it have any salt bridges or disulfide bonds? How does it do whatever it's supposed to do? Its biochemical role. What's its chemistry, or is it just a structural protein? How's it regulated? All right. We'll talk about regulation quite a bit later. How does it interact with other ones? Does it bind to other particular proteins? How's it related to other proteins? Is it in a particular family? Is it similar to other proteins, similar to other organisms' proteins? Where is it? Is it in the cell surface, in the ribosome? Does it have its own little specialized compartment that's located to extracellular matrix? And we can even ask what are its physicochemical properties. So that's where we start getting all sorts of craziness in here. So we got a lot of stuff we can try and ask about proteins. Some of those will be easy to answer, and some of those, like, well, hell if I know. So try and figure it out. All right. So a lot of times when we're trying to study proteins, we're going to have to try and purify them. And this was actually, I always like to tell the story about the lab that I first ran, where I would go to Disher Meats over near Yadkinville and ask them for a beef heart. And they'd give me like about a heart, almost the size of my head, from a cow. And then we would spend 6 to 8 weeks chopping it up and isolating a particular protein from it. Right? Our lactose dehydrogenase, because that's a very common protein in muscle to help deal with the buildup of lactic acid. And we just start working on trying to separate it out and purify. Right? So how do we get that protein out of this mixture of other proteins, other fat and stuff like that? One of the challenges was when you cut up heart muscle, you got a lot of ligament in there, you got a lot of little stuff, goop that will kind of give you a solution that is they're not very homogeneous. So you want to try and get just the actual muscle, the muscle meat. I don't know if you guys like eat steak, and like, for example, my son enjoys the fatty parts of meat, and like, I can't stand it. You know, if you get like a cheap steak, you get have lots of gristle in there as well too. So it's like I just want meat, pure meat, you know, pure protein. Okay. There becomes kind of
No, that's the opposite, man.
all right. So if we look at the sequence and arrangement in there, we can, you know, kind of figure out, is this a polar protein, or is it hydrophobic, or what's that? Some polypeptides will bind to particular targets when we have an interaction. Maybe we'll have, say, a glucose binding protein, and we can actually use a column that has glucose attached to the stationary phase, and we can pass a mixture through there, and ideally our protein will bind to that stationary phase. We can wash all the other stuff away, and then we can elute that off, and we'll talk about that later today, or maybe on Thursday. I think today. Yep, we're getting into today. All right. So if we chop up our beef heart and stick it in a blender, wasn't there like a song about that from the early 90s? Some cheesy, I don't know, California band. Keep mine. Yeah, heart in a blender, something like that. Probably should have played that music when we were doing that lab. But that lab broke down, so we're not going to worry about that. Again, this is going to be awesome on Zoom. Okay. For those of you watching later, I should go in and like edit out all the stuff that I don't speak, but then you'd have actually like 20-minute lectures, right? It'd be longer than that. Anyway, we can separate out proteins based on their charge. If we can figure out the isoelectric point, okay, well, we can figure out whether we want to make it negative or positive. So we change the pH of our solution, and we can have our proteins migrate in different ways. We could also isolate them based on size. If I want to separate titin from cytochrome C, that would actually be really easy, because we can come up with what's called a molecular sieving technique, which we'll talk about here in a little bit. We can look at solubility as well, affinity for ligand, hydrophobicity, thermal stability. There's lots of different ways that we can manipulate a solution to try and separate out this stuff. And if you have a very small concentration of a protein in your sample, you might need to use 2 or 3 different methods to gently separate it and get as much of that small protein as you can. In most cases, we'll talk about some form of chromatography. So if you guys recall from organic chemistry, you probably did thin layer chromatography, right? Where you plot like different compounds on a silica plate and see how far they'd migrate up with their capillary action, right? So we're gonna do something somewhat similar. Basic premise in there, our chromatography. We're gonna have a solid phase and a mobile phase, and we're gonna try and change the properties of our phases so that we can get our proteins to separate, okay? So the basic idea here at column chromatography, we take this mixture of proteins, put them on the column, they interact with the stationary phase in different ways. So in this case, our green one reacts less with that phase, so it passes through faster. So we get this, band, this green band for protein C. Our B maybe has an intermediate interaction, so we get this band for B. And our A interacts the most, so it migrates the slowest. So then we can collect our different samples and fractions. And we can see, oh, I'm collecting the C. And then you have different detectors in here. You can run these up on gels to figure them out and stuff like that. So our basic idea of those, we pass through the column to help separate this out. Just like if you're straining spaghetti, put it in the strainer, the water passes through, the spaghetti stays behind, you've separated out that solution, right? Getting close to lunchtime, must be hungry. Okay. All right, so there's 3 major ways that we can separate these. Probably the most common, the oldest most common one is ion exchange, okay, often abbreviated IEX, and this is going to be based on the charge. So of course, if we change the pH of our solution, we can change the charge of our protein in there, because we'll either be protonating or deprotonating our proteins. Okay? So in this case, if we have a negatively charged stationary phase in here, a bunch of negative ions in there, our positive proteins will hang out. They'll be attracted to the stationary phase, so they'll migrate more slowly. Our negatively charged ones will be repelled, so they'll go through there faster. If we've got a big negative charge, they're going to zip through there. Got a small negative charge, they're going to move through there. They're not going to hang around. They're not going to loiter, but they're going to go. A small positive charge, they'll be like, hey, look at this cool thing in the window. They'll pause for a little bit, but they'll kind of wash through. And our large positive charge ideally will stick to the protein, to the stationary phase. And then we can change the pH of our wash buffer to elute that off, ideally. So basically looking at that pH. This is why our isoelectric point becomes important. Maybe we'll put them on in a pH of 5 and elute them in a pH of 8. Completely change the characteristics so that it doesn't stick anymore. All right. Does that make sense? Too bad. Yeah, we manipulate the ionization state of our protein to try and get rid of the ones that don't match. Of course, if we have like a huge number of proteins in there, you might have one protein that ends up at a certain pH being plus 20. You have another one, a different protein that ends up being like plus 18. Like, oh crap. So they're both probably going to elute the same. Okay? But you can get rid of a whole bunch of other proteins in there that are different. So ion exchange now is sometimes used as like the first step to just get rid of all the stuff that's really different from what you're looking at. Let me skip that one and come back to that one. The next one I like to talk about is similar to ion exchange, but it's more specific. And I like this because you can do this in one step. Sometimes, depends on how concentrated you're. And affinity chromatography, sometimes abbreviated AC, you basically find a protein that binds to a particular ligand. Like maybe it's an enzyme that's going to bind to something and perform an action on it, or maybe it's just like a carrier protein in there. So you can actually attach your ligand to the stationary phase, and then your protein is going to come and bind to it. Right? And depending on the specificity, for example, if you wanted to take albumin from like egg whites, you can find what it binds, put in there. It's going to bind really tightly. Then you're going to have to figure out how you're going to elute it as well too. But the idea is you take your mixture, pump it in there, your protein of interest binds to the stationary phase, everything else passes through. Okay? Now some things, if there are several proteins that'll bind NADH or ATP, so that might not be the best choice for a stationary phase, but that can also be a good start if you have a bunch of huge mixture there. But you can get rid of a lot of this stuff. If you have a really specific binding partner, you can sometimes do this in one step. You take this whole cell extract, pass it through there, everything comes off except your protein of interest, and then you can elute it. You'll have your elution buffer. In your elution buffer, there's a couple different ways we can do this. Theoretically, the way they say is to make a mixture of whatever the ligand is, make a high concentration, usually like 1 molar ligand, and then send that through there, and it'll wash off all that protein that was bound. Right? Because now you're basically when your protein comes on there first, there's like, oh, one thing I can grab onto. So your proteins grab onto it. Okay? When you send your elution buffer, like, oh, there's lots of these things floating free in solution, so they come down with them bound in solution, and you can isolate them out that way. That's one way to elute, but it turns out there's another way to elute that is often cheaper and easier to do. You can make a high salt concentration compound. So a lot of times our elution buffer will be whatever our loading buffer is plus 1 molar sodium chloride. So why does that work? Okay, with 1 molar sodium chloride, you got a lot of positive ions, a lot of negative ions in there. So you have this electrostatic disruption. And that high concentration of dissolved ions is going to interrupt the ability to bind in here, cause them to fall off. And then you can just dialyze that to get rid of the NaCl solution. So that's a way where you don't have to worry about how do you get the target off of the protein. You just disrupt its ability to bind that target when your elution buffer, and you never have to worry about it. Okay. That's 2 different ways in the elute. That also makes a good question for the exam. What else did I want to say about that? Yes, okay. So we show this on a column, but actually one of my favorite techniques, and this is like 20 years ago now, so this is super common now. You know, I was working with some type of cell, some bacteria that we grow up, and you know, you could make a whole cell extract and pass it through the column and then elute it, but we ended up using magnetic beads. So rather than have a stationary phase in a column, I just had like a little tube, and I would take my whole cell extract and put it in there. My little beads were much like the stationary phase in here. They're little beads that would have our ligand of interest, our ligand that would bind to our protein of interest attached to the beads. So then I'd let that mix for like 5 minutes, and then I'd spin it down. All the beads would go to the bottom. I could pipette off the supernatant and get rid of all this crap here. And then I could add a few drops of my elution buffer and then let that incubate for 5 minutes. That would pull off all the protein. I could spin the beads down. When I pipette off my supernatant, I now have purified protein in my supernatant. And that was like a 26-minute protein purification. I mean, it was super fast. It was awesome. It's too bad about that lab. I'll tell you my horrible postdoc story at some point. But it was a great technique, and I love that. And it used the same principle. It was just super fast, which was great because when I did my graduate work, I actually had 3 columns that I would purify a particular protein off of, and whew, that took a long time. All right, but that's the affinity chromatography method. So even if you're not actually doing column chromatography, you can still use this method to purify. There's a third column purification that can be useful. Right, I should switch these slides around. So I like to talk about this one third. And this one is called size exclusion chromatography, sometimes called molecular sieving, sometimes called, there's another name for it too, but I forget what it is. Gel filtration. Okay. And basically what we do here is we use a solid phase that are little beads that have various size holes and tunnels and pits in them. Okay. And the idea here is we're altering the path length that a molecule can follow. So if we have a really large protein, it can't fit in any of these tunnels or holes or stuff like that. So it's got one path. It's like the big guy who's got to go straight down the road and it's got to leave. So our large proteins come off first because they have a shorter path that they can travel. We're using a lot of statistics in here. We have, say, a medium-sized protein. Our medium-sized protein can fit in some of the tunnels, can fit in some little nodules in there. So, you know, it can meander randomly a larger path. So it'll come off a little bit later because it can fit in more places. A small molecule will fit in all these tunnels. So it's really got a really long distance that it could theoretically travel. So it's not actually attracted to the stable phase, but it just finds, oh, where's this little path go? Oh, we're going to go over here. And so it kind of wanders around in circles a bit. And our smallest proteins come off last because they have a longer path that they can travel. Okay? So size exclusion is a good way, if you know the size of your protein, to separate it out. And the key is larger proteins come off first, smaller proteins come off last. Make sense? Not awful, right? Okay, biochem's not that bad. It's just there's so much of it. All right. Okay. So once we've got a purified solution, what do we do with it? Drink it. Okay. Probably not. But we can analyze it, and one of the ways to do this is to just kind of check it by running a gel. So and then maybe treating with antibodies. We can do Western blot in there as well. But we're going to do some gel electrophoresis. So we're going to basically, much like our DNA and RNA agarose gels, I assume probably most everybody's done an agarose gel in biology, right? Maybe, but yeah, okay, yeah. So our polyacrylamide gel, same principle. We're going to make this gel matrix, all right? And then we can, we can figure out how stiff and how complex the crosslinks are in there. So if we have a bunch of tiny proteins, we can put in a lot more crosslinks to help separate them out. If we just have big proteins, we can use less acrylamide, fewer crosslinks, but the number of crosslinks we make in this gel, I always think of like the sit-in video games from the arcades when I was a kid. You're like, what? But yeah, you have a spaceship and you're trying to fly through these places, and all these things that are crossed. So the more cross links you have, the harder it is to get through. So that will slow down our larger proteins. They can't fit as well. Our small proteins, like, our X-wings that are flying into Death Star, they're fine. Should edit that one out. Anyway, but our gel matrix will, again, separate them based on the size and shape. Okay. Most common, we're going to use polyacrylamide. So we'll call this polyacrylamide gel electrophoresis, or PAGE for short. Okay. And I think we're going to see an example of this in Labster. I'm still nervously going, okay, Labster, come on. Let's do that. But we basically load our samples in here. We'll have some type of dye so that we can see it, see where it's loaded in there. Depending, I know I talked about pipetting. For some particular types of PAGE, you may have different shaped wells, and you'll have this long, feathery looking pipette tip to help get them in there. But for the most part, you can kind of load these in here. You have your sample wells, and then we'll apply an electric field over here, and that will cause our protein to migrate. We'll keep track of this dye front, which will slide down first. When our dye front gets near the bottom of the gel, the dye is very small, so typically all our proteins will be behind that. And then we can stain it and kind of look at where our different bands are to see our different proteins. Okay. So one of the issues we run into is our proteins fold, and they have different shapes. So if we want to separate out proteins just based on their molecular weight, we need to unfold them. So we'll treat them with something that will facilitate that unfolding. Most commonly, we use the sodium dodecyl sulfate. You can see this is like essentially a type of detergent, but it's got this shape in here. It's got this lovely little charge. So the SDS will form micelles that'll bind to the proteins, and it'll actually kind of pull them apart and unfold them so that you'll get long linear proteins that are surrounded by these negatively charged SDS ions. Okay? So we'll have this uniform negative charge. The bigger the protein is, the bigger the negative charge it'll have. The negative shape won't matter. So our rate of movement will only depend on size. Small proteins will move faster, large proteins will move slower. Right? And we'll end up with maybe something that looks like this. Okay? So this is our typical purification check. We have size markers over here on the far left. So you can see we just know what size these are. We'll put these bands in here. We might have uninduced cells. So we just take a protein extract from cells that haven't been treated to make this one particular protein. In this case, induced cells are stimulated to produce this one particular protein. Okay. So we can see this difference here. Everything looks fairly similar, except we have this extra band here. If we take this soluble crude extract, this band becomes so big it kind of overwhelms stuff. But you can see that crude extract has tons of other proteins in here. So I precipitate it out with ammonium sulfate. This is a good way to get proteins to precipitate. And you can see you've got like your protein of interest and maybe another one in there. Okay, so there's another band down there. So I do maybe ion exchange. I get one band. Maybe there's a shadow there, maybe not. I do a cation exchange. I definitely get one band there. So I compare this to maybe like a sample of purified protein that I bought from a company, and I can see, hey, it's the same size. There's nothing else in here. So our ion exchange worked pretty well to purify, and this happens to be the Rec-A protein from E. coli. All right. So there's another neat thing we can do. A lot of times, like most of the stuff we're going to work with, it's going to be a protein that we know about. We're going to know its molecular weight. We'll often know its sequence and stuff like that. But if we have an unknown protein, we can figure out its molecular weight. All right. And the way we do this is a way that we never actually do this in lab. I keep thinking I'm going to do this in lab one day, but it requires a little graphing. Okay. So we have the top of our gel. And actually, this is not really true, because normally what we do is we have a stacking gel and then a running gel. And our stacking gel is less dense, so that our protein will get into it really fast. And then we hit the main gel. So we would actually take a look at our main gel and calculate the distance that our proteins have run. So I just measure this. I basically just take a measuring device and see how far this has gone. Okay, and I compare. I basically make a plot of my known markers that I have here and how far they've run. Okay? And what I'm going to do is I'll plot the log value of the molecular weight versus the distance they've migrated down here. So you can see the log of myosin. It's going to be relatively large, but it's not going to move very far. Okay? Log of this versus the distance it's gone. So I end up making this standard curve, okay, based on my size markers. And ideally, this would be in a perfect little straight line here, and I can get a line equation. Okay? My unknown protein, okay, well, I don't know what its mass, its molar mass, but I can measure how far it migrated down there. I can plug that into my line equation to get the log value of the molar mass. I take the anti-log of that, and it'll give me the actual weight.
Waxing Crunchwrap Supreme.
Now, no one ever really does that.
A bean burrito.
Even when they're talking about
And chips and cheese.
Hold on here.
Just one order of chips and cheese. That's it. Any taco sauce? No.
Where's my mute? All right. So.
That's it.
All right, everything's correct on your tray?
Yes, ma'am.
Any taco sauce for you today?
No.
All right, it's 661.
Oh, I'm ready yesterday.
I gotta find this in here. Oh well. All right. Well, anyway. I hope that means tacos for everybody. Anyway, so what we do is we can figure that out. But how do we really do this? All right. Most of the time we eyeball it. Okay. So if I look at this as 31 and 45, I can say this is maybe a little closer to this one than that. I'd call this maybe 39, 40,000 molecular weight. Most of the time, that's going to be in the ballpark. If I know I'm looking for something that has a molar mass of 38,000, I'll say, yep, that's probably it. I'm not going to measure all this out, go through these calculations and plug all that stuff in there and solve a line equation and all. Only if we're really working with an unknown protein is that what we end up doing. So that's not hideous, but it's just functionally what we really do in lab. Often just kind of eyeball it. Okay, now I can get back to this. Hey, got it, figured it out. Now I just need to be able to do that fast. Okay. Like that. And of course, everything changes from my screen, but not for your screen there. Okay. So that's how we actually do that. Okay. So our SDS page gels are one way to do that. And again, we're linearizing it, so we're only looking at size. We can also use our polyacrylamide gels to figure out the isoelectric point of a protein as well too. Okay? So what we can do is we can make our gel so that we have a gradient of pH. In this example, our pH might go from 9 to 3. We inject our sample at kind of one end of our gel up here, run our electric field across here. We see our decreasing PI as we go across here, as we change our pH. So our proteins are going to migrate. We could let this run forever, for like a month. But eventually, our proteins will reach a pH where their charge is 0, and they'll just hang out there. Because if their charge is 0, they're not going to move either way in the electric field. So they'll just kind of hang out there as these little blobs. Okay, so you can separate the proteins based on their isoelectric point. Okay? Of course, you might have a bunch of different proteins that all have very similar isoelectric points. Maybe like pH 3.8 and pH 3.9. So how do you separate those out? You can actually combine these two techniques. So you do isoelectric focusing first, and then so you get your PI is going across this way. Then you apply an electric field in SDS this way to separate them based on their size. So this is called a two-dimensional gel electrophoresis. If you have a sample that has a ton of proteins in there, this is what you end up with. And it's a big mess. It's like a Jackson Pollock painting. Oh, look, timely art references. They're from like 70 years ago. All right, I guess if you're the first one to throw ink at a canvas, you become famous. Okay, but we have this technique that can help us identify particular proteins, especially if we have like a big messy extract. Okay. All right. We can also look at the way our proteins absorb light. Okay. If we look at our aromatic amino acids, so we got those ring structures, right? We got a lot of conjugated p orbitals, pi bonds up there. So they're going to absorb UV light. Our proteins are typically going to absorb somewhere around 275 to 280. So we're looking primarily at tryptophan and tyrosine, phenylalanine, in there a little bit. But typically we're looking at 275 to 280. Tryptophan and tyrosine are the strongest ones. And of course, we can look at our absorbance being directly proportional to our concentration. Right? And of course, our A equals epsilon CL. It's just L is our path length. It's funny, I always joke about the path length is pretty much always 1 centimeter, except there was one time where I encountered a machine that had like a 2 centimeter cuvette. So it had a 2 centimeter path length there. Our epsilon is just this kind of fudge factor, extinction coefficient for each individual protein stuff, whatever you're putting in there. But anyway, if we look at this, if we alter our wavelength, we can see our absorption here. See, phenylalanine, not very good chromophore, but tyrosine and tryptophan have these nice, very distinguishable peaks somewhere around 280. Okay, so typically if we're looking for protein, take a solution, stick it in a spectrometer, look at 280, and you get these nice big peaks in there. You're like, hey, we got proteins here. Unless you have some weird protein that doesn't have any tyrosine or tryptophan. You always got to look for the outliers. Sorry, I watched way too much X-Files when I was younger. In fact, my first year of grad school, I made sure my schedule was clear so I could go home. This is when some TV channel was running them back to back for 2 hours every afternoon. So from like 1 to 3, I'd go home and watch X-Files as a grad student. Terrible. All right. Okay. Definitely got to cut that out of the Zoom lecture. I don't even bother. You get what you get. It's performance art. Okay. We can also talk about more characteristics of our proteins. It's like, I feel like I should be on an infomercial, but wait, there's more. It slices, it dices. Talk about dicer. Anyway, the specific activity is going to tell you how pure your protein is. All right. So if you have a completely pure sample and it performs some function, maybe it cleaves a certain molecule into 2 other things, you can measure the rate at which it does that, and that would be like your best activity. So if you have a protein that you're trying to purify from a solution of proteins, if you're at a lower concentration, you're going to have a lesser specific activity. So we can use specific activity to measure how good our purification is, right? Because again, if we have a whole bunch of different proteins in this really complex mixture, we might have a lot of stuff to try and get out of there. We might know how much protein is in there, because again, we can look at that absorbance value, but not all of that would be our protein of interest. So every time we do a purification step, we can measure specific activity to figure out if we're actually continually increasing the purification of our prep. Okay, so we might look at, in this case, binding insulin. The fraction that binds insulin best will contain the most of the protein of interest. Okay, and this is our activity. If we take a ratio of the total protein concentration to the activity, that's our specific activity. So for example, oh, come on, there we go. We might start with a solution that has all these different proteins in there, okay? And we're interested in, it'd be better if we had, I was gonna say maybe the blue ones, but it seems like we got rid of most of the blue ones. Maybe the red ones, yeah, we're trying to get the red ones or something like that. So we go through around the purification, and we get rid of most of this stuff, but we can see, our ratio of red balls to the rest of them has gotten higher in between this step, so our specific activity is going to increase. So even though we have less total protein, we have more of the red ones here. We have more of our protein of interest. Ideally, we'll get to a point where we'd have a little flask with nothing but the red balls in there. Okay. All right. Good times, right? Yeah. All right. All right. We might actually. Woohoo! See, there's a part of me that's thinking we're moving along at a good clip here. I got to switch from having 3 one-hour classes to 2 hour and 15-minute classes. It's thrown off my timing a little bit, but I will not give you guys an exam the day after Labor Day, so it'll be next Thursday. But we might do some new stuff on this Thursday, do a review session on Tuesday, and have our exam a week from Thursday. Yeah. All right. So our protein sequencing, how do we do this? All right. Now this gets to a point where this is more informational stuff. I'm not going to go very deep into this because I like a third method of protein sequencing, but that requires genetic knowledge. So our protein sequence, we got to figure out what the sequence is of our protein. What are the amino acids that are hooked together? What order are they hooked together? So our actual sequence comes from the DNA sequence, or more specifically the mRNA sequence, or more specifically the exon sequence that's properly associated there. But we can find that mRNA sequence and come up with a protein that way. Sometimes though, you get different splicing in there. So we might want to sequence our mature protein. So there are 2 methods. The Edmond degradation, which is a classical method. It's slow, but it's really accurate. And we basically have successive rounds of we modify the end. And we figure out what's on the end, cleave that off there, and then we do it again, figure out what's on that end, cleave it off. So it's kind of like this one step over and over again. Okay, so we modify, cleave, and identify. It's best to identify a protein with a known sequence because we can figure out what the sequence is in there. Mass spec, particularly looking at like MALDI-TOF and all that good stuff in there, we can really identify the mass of a peptide, and then we can use that to map it out and figure out the amino acid sequence. The good thing about mass spectrometry is we can look at post-translational modifications and figure those out there as well too. If we take a brief glance at both of these, now I remember I think we had like 2 weeks on MALDI in my analytical chemistry when I was an undergrad. But if we look at the Edmond degradation, since we have it here, here's our protein, we have our long polypeptide, and then we treat it with this phenylisothiocyanate, which will bind to the amino terminus in there. Okay. So we end up with something like this, and then we're going to cleave off the end here. Very low pH. We're cleaving this off the end. We come up with this, and then we figure out what this is. We can identify this. We have this amino acid residue in there. All right. And then we get rid of this and repeat this again. Okay, so we purify this, recycle it through there, and keep going. So we're constantly making these phenylthiohydantoin derivatives in there, and we can just figure out what that R group is based on that. Okay. So we're always making a nice little cleaved off action there. Like I say, it's slow, it's step by step. Just cut off a piece, figure out what that is, cut off the next piece, figure out what that is, cut off the next piece, and keep going. Mass spec, fire them through there into our mass spectrometer. You get all these lovely little peaks. You guys have to assign peaks in, what would this be? What chemistry do you do, MALDI, in which class? Organic? Okay, yeah. I remember doing lots of NMR peaks, and that was fine. MALDI was pain. But again, separate all these in there. We get all these nice little fragments, give us all these little individual peaks, and we kind of put them back together. Analytically speaking, this is like one of my favorite techniques when I was an undergrad, which we don't do anymore because there's more efficient ways to do it. But looking at DNA sequencing, restriction enzyme analysis. You just take different restriction enzymes and cut a segment of DNA into small pieces, run it out on gel, figure out what the sizes were, and then kind of put the puzzle together, figure out what the sequence was. We're essentially doing the same thing here, except instead of doing like a 100-piece jigsaw puzzle, we're doing a 5,000-piece jigsaw puzzle. So yay for computing power. All right. Again, much faster than our Edmond degradation. And assuming that we have a good library of data for all of our proteins and their post-translational modifications that they can have, we can resolve this much quicker than our Edmond degradation. Although actually Edmond degradation sometimes is not bad because you can have robotic, basically have robots to do the whole thing for you. So you just get it set up, make sure the reservoirs are filled, and it'll sequence for you. All right, so why do we want to sequence them? Just beside wanting to know what the sequence is. If we know the sequence, we can understand relationships between proteins, between organisms, between organs. We can look at analyzing protein families to help figure out evolutionary history. We can help understand disease as well too, and we'll kind of look at that later on in the next chapter. All right. So that's it for this chapter. So we talk about our basic peptides and the 20 amino acids, which you should have your flashcards and be memorizing those, or whatever, quizzing yourself and all that good stuff in there. We looked at some of the functions of them, what they do. We can talk about how to separate out proteins and peptides, our ionization properties, our isoelectric point. So there's a fair amount in this chapter. And this is also kind of like a nice, good little stopping point to not overwhelm anybody. Introductory. So our first 3 chapters are going to be in our first exam. The first exam, yeah, yes. There is a study guide. I believe it's posted in Canvas. I'll check and make sure it's visible. I know sometimes things change when they do updates. Right. Yeah. So as far as the first exam goes, it's not awful. It's not great. It's the first exam, so everybody's kind of getting used to me and the way I ask questions. I'm also kind of trying to determine how I want to do the exam. I want to do it fully online, or we'll see. We'll see. I'll know more by next couple days how I'm going to do that. Yeah. That's it. Yes. Yeah. Yep. We'll probably peek ahead into chapter 4. The interesting thing is, now we start linking up our chapters. So each chapter here has been something pretty different, But chapter 3 and chapter 4 just flow into each other to the point where sometimes I mix up what's in there. I have to be careful what questions I want to ask on the exam. Like, oh, nope, can't ask that yet. But we're going to see that peptide bond again and again and again. And then that flows into chapter 5 and 6. And then we switch gears after that because everybody will want to break after chapter 6. All right. Okay, so let me stop this. And that's it. Yeah, we'll let you guys out a couple minutes early today. Woo! Good times. Okay. If I do this, this will help.